actually, since we're recording, let me just go through this one more time verbally since I'm recording this. Attendance, very quickly. Sorry, guys. Abigail is here. Actually, it's quicker to say he's not here. Andrea is not here. Cooper is not here. Reinhardt is not here. Talbot is not here. Anna is not here. Galloway is not here. Cook is not here. Jones is not here. Tristan's not here. Hobson's not here. Ray is not here. And that's it. All right. So recording, got that up there. Let's pull this up. Download this, and then we'll open it. So again, while this is downloading, I want to remind you, this week is just a pre-lab lecture. This, I'm going to tell you everything you know about how to do this lab and how to write a lab report. And then when we get here next week, we're going to hit the ground running and get right to work. This is one of those longer labs, and I know no one wants to stay here longer than they have to. So again, hopefully we'll hit the ground running, and you'll know what you're doing next week. That being said, if you do forget something that I said today and you have questions next week, obviously feel free to ask. It's not like you need to memorize this stuff. That's a little bit hard to see, but we'll try. All right, here we go. Photosynthesis respiration lab. So today's lab grade, well, this is actually old. Let me read this anyway and see. I might need to change it. Today's lab grade is based on attendance. That's true. Not the pre-lab. That was from last semester. So for you who are here, this week's lab, you got your full 20 points just by being here. Unless you leave early, then you're going to lose some points. But you did it. You got the full 20 points for this week's lab. Next week's lab is going to be normal. You have the pre-lab, and then you're actually going to do the lab. So the grading for next week will be just as you're used to. So again, forget what that says up there. You're here. You got your full 20 points. For some odd reason, you want to come on Wednesday and sit through the same lecture, and you get a little extra credit. But anyway, here's some questions that we're going to answer in this lab. The first one is, do photosynthetic organisms photosynthesize faster and more intense light? So basically, as we give plants more light, well, there's other photosynthetic organisms, but anyway, if we give something more light, which I'm going to do less light so we can see this better, does it photosynthesize faster? What do you guys think? If we give something more light, do you think it'll photosynthesize faster? I would think in general the answer is yes, right? Because you have to have light for photosynthesis. But there are some exceptions. We'll talk about those in lecture, and hopefully you'll get some data next week. The other question we're going to answer is, do photosynthetic organisms respire? So not photosynthesis, but respiration. Do they do respiration when light is absent, right? So when they're in the dark, do they do respiration, not photosynthesis? Any guesses to that? Any hypotheses? Anybody want to go out on a limb and see if you're correct or not? Well, these beads of algae, will they go through respiration in the dark? What do you guys think? Little tiny beads. No guesses? Doesn't matter. We'll see. My guess is yes, but we'll see what happens. The next one is, do photosynthetic organisms photosynthesize and respire faster at warmer temperatures? So in other words, we're going to do a temperature experiment, see if temperature affects the rate of photosynthesis and respiration. And then the last one we're going to ask is, do photosynthetic organisms photosynthesize at different rates with different colors of light, basically? When we say different wavelengths of light, that's what I mean. If you can see here, you guys are going to be dealing with blue lights next week, and green lights, red lights, white lights. If I get extra lights, different colors, we'll use those too. But we're going to see if light matters. So those are the questions that we're going to answer in this lab. So how do we answer these questions, right? That's what we're talking about next. How do we measure the rate of photosynthesis and respiration? Does anybody remember, and I don't blame you if you did, don't, because we only brought it up today, but does anybody remember the equation for photosynthesis? Respiration. What are we making and or what do we use? What do we use in respiration? What do we produce in respiration? Does anybody remember? Let's just start with what do we use? For respiration, and yeah, we're talking about cellular respiration, but what about you? When you respire, what are you trying to get in? Start with that. That's the easy one. Why are you respiring right now? Because none of you look like you're holding your breath. Why are you respiring? What are you after? I saw you breathe in. What are you after? Oxygen. Yay. Good. Why did you breathe out? What are you getting rid of? Okay, good. Now, if you were to breathe on a mirror, what would happen? It would clog up. That's not carbon dioxide, so what else must be coming out of your mouth? What is it? What would make a mirror fog up? Water, good. And what's the whole purpose? All right, so we know we're producing carbon dioxide and water. What else are we making? Like, what's the purpose? What are we trying to make? Yeah, we produce water and we produce carbon dioxide, but what are we after? What are we trying to produce in respiration? Like cellular respiration. Why would we go through it? We talked about it today. What's that one molecule that we're trying to produce? A bunch of. You're off. We'll use it. Great. We're going to come back to that. So keep that in mind. Glucose is definitely part of this equation. But what are we trying to make in cellular respiration? What's the big thing we're after? We happen to make some carbon dioxide as a byproduct. We happen to make some water as a byproduct. But what are your cells trying to make? It's a really high energy molecule. That's all right. I only told you today. You haven't had time to study. ATP. That's what we're after. We're trying to make ATP. Anyway, like someone said, we're trying to make ATP. We're making carbon dioxide and water in the process. We're breaking something down in the presence of oxygen to do this. What was that thing? Someone said it. Good. Glucose. C6H12O6. Right. So that's the equation. That is respiration. And just so you know, photosynthesis. We'll call it like this. That's respiration. Photosynthesis is just the other way. Now, this is not trivial information. This comes back to this. It's a long-winded way of getting to what I was trying to say. How can we measure the rate of photosynthesis and respiration? So if we had all semester to do this, we could grow a plant, right? We could grow a plant in the red light, a plant in the green light, a plant in the white light, for example, and just measure the height, right? Measure the height every week. How much did it grow? Or the number of leaves, or its weight, or its, its stem circumference. There's so many ways you can measure like how much a plant grows in a semester. But we don't have a semester. We have about 2 hours. So here we go. There's the photosynthesis equation, right? Like I already told you, water plus carbon dioxide gives us glucose and oxygen. The opposite is true of respiration. Respiration, we break down glucose in the presence of oxygen. That gives us carbon dioxide and water. So this alone gives us a lot of options, right? If we wanted to measure photosynthesis, for example, if we could, if the amount of water increased or decreased, right, we know photosynthesis is happening. Does that make sense? Because photosynthesis uses water. So if the amount of water decreases, we're like, oh, photosynthesis is happening. It also uses carbon dioxide, right? So if carbon dioxide is going down, we must know that photosynthesis is happening because it's using carbon dioxide. Does that make sense? All right. Photosynthesis produces glucose. So if somehow we can measure the levels of glucose, if the levels of glucose were increasing, we would know photosynthesis is happening. Same thing here. If we can measure the levels of oxygen, we would know photosynthesis is happening if oxygen is increasing because photosynthesis produces oxygen. Does all that make sense? Really quickly, the opposite is true of respiration. Respiration uses glucose, so if we could measure glucose levels and they were going down, respiration must be happening. If oxygen levels are going down, respiration must be happening because respiration uses oxygen. If carbon dioxide levels are going up, respiration must be happening because respiration produces carbon dioxide. And finally, if water levels were going up, respiration must be happening because water is produced, right? So if we can measure any of those things, we can measure photosynthesis and respiration. And the good news is we can measure those things. As some of you already know, because you've written papers about this, if you mix carbon dioxide and water, you get an acid. It's called carbonic acid. The type of acid doesn't matter. What you definitely need to know is you need to describe this in your lab report. If you mix carbon dioxide and water, you make acid, right? So there we go. That's all we really need to measure photosynthesis and respiration because photosynthesis uses carbon dioxide. Therefore, right, since photosynthesis reduces the amount of carbon dioxide, that would reduce the amount of acid in the solution. Therefore, the pH goes up. Does that make sense? It's really important. I can't go forward until you make sure you understand that. When something is photosynthesizing, it is using carbon dioxide. Therefore, it is taking carbon dioxide out of the solution, which means there's also less acid in the solution, which means the pH is going to increase because things are getting less acidic. Is everybody with me so far? Okay, good. Keep that in mind. pH goes up. That means photosynthesis is happening. Excuse me. Fix this. Anyway, back to this. And the opposite is true of respiration. If respiration is happening, it is producing carbon dioxide. Therefore, the carbon dioxide levels are increasing. Therefore, the levels of acid are increasing, right? We're getting more acid. Therefore, the pH is going to decrease. So all that makes sense. So that's your key. What we have here, and I'm glad you brought them now, because I can physically show you. We've got these little things that have algae beads in them. A little hard to see. Actually, you can pass one around once I'm done. These have algae beads in them. And you can see that water is like a, I don't know, orangish color. I'm really bad at describing colors. But anyway, it's a particular color. And it will change. Because remember when we did experiments with pH, we had pH indicator in there, and it changed depending on what the pH was? That's what's in here. So we can see the changes in pH. We can see if the pH goes up. We can see if the pH goes down. You pass that along as we talk. Use to snake it around. So there we go. That's the basics. Make sure you understand that before you start worrying about any of the details in the lab. If you don't understand these basics, then all those other stupid little details mean nothing. So how are we going to do this? You're going to have one of these cuvettes, which are getting passed around right now, that's filled with pH indicator. Like I already said, it's this particular color that changes depending on the pH, and algae beads. Every 5 minutes, you're going to check the cuvette, right? So that thing going around, you're going to check it. I know that's hard to see, but you're going to have a little strip that looks like this. So maybe you did it already. Did you grab it? Okay. We'll need that for next week. So this is perfect. So yeah, you have this little strip like you did for lab 2. You compare the color of that thing to the strip and determine the pH. Does that make sense? Pretty simple. You're just going to do that every 5 minutes. Let me ask you this, though, and you're going to put it in your table, which is kind of hard to see right there. But why do it every 5 minutes? Why don't we just take the pH at the beginning and then take it again 45 minutes later? Why do we need it every 5 minutes? Any guesses? All right, let's see. Let's say we have one in the green, one in the red. I like to use the ones for color as an example. Let me say 5 minutes, down 5, 10, 15, 20, 25, 30, 35, 40, 45. All right. Let's say this one starts off at a pH of 4. This one also starts out from a pH of 4. Photosynthesis, so it goes up. Let's say this one's at 7. This one also ends up at 7. 45 minutes later, here's what happened. So which one does better for photosynthesis? Green light or red light? Based on this data. Can't tell. Perfect. Good. Because it all started at the same place and ended at the same place, right? But what if it does this? What if green goes, I don't know, 4, 4, 4, 5, 5, 5, 5, 6, 7, right? And red goes 4, 5, 6, 7. Now, which one do you think is better for respiration? Red light or green light? Red, right? Because it got there really quickly. And as a matter of fact, we could tell you the list and maybe get a story from the data. I would say perhaps 7 for some reason is the highest that's going to go. For whatever reason, maybe that's our limit. There is a limit to the pH. I don't know what it is. I mean, we know 14 is the highest, but that particular vial, this thing of algae beads, it's only going to get so high. I don't know how high, but it only gets so high before it maxes out. But there we go. So that's why we take it every 5 minutes, because you can't look at just the beginning and the end. Maybe you can, but you might have a situation in your data in which you need to look at the whole picture. Yeah, just put that in there. I'll take care of it. Thank you. So any questions about that? All right, pretty simple stuff. So again, those are the questions we're gonna answer. Let's talk about each one individually very quickly. Do photosynthetic organisms photosynthesize faster and more intense light? That's the first experiment. Some of you are going to do the first experiment. Some of you are going to do the other ones. So you're going to have one in the dark. You're going to have one close to a light. And you're going to have one even closer to the light. And we might even have an additional one. The point is we're going to have a bunch of them. We'll have a light on. We'll assume it was a white light. We'll have a white light like this. And we'll just have a bunch of them. Like one's going to be really close. One's going to be a little bit farther away. One's going to be really far away, depending on how many we get. We might get extra, in which case we'll just do a bunch extra. But at least we'll have 3. Does that make sense? All right, so again, what we're looking for in this case is for the pH to increase. Because if increase, right? All right. I'm sorry. No, that is right. Yeah, the one in the dark. Sorry. There's also going to be one in the dark. That's our control. The dark one is not getting any light. Therefore, in theory, it should be doing respiration, not photosynthesis. Therefore, that's the only one that should be going down. Right? The rest of them, we should expect an increase in pH, meaning the photosynthesis is happening. Does that make sense to everybody? All right. All right. So this is the other one. That's experiment 1. Experiment 2, which some of you will do and some of you won't. You're going to have 2 cuvettes. You're going to have one in the light, one in the dark. That one's really easy. The one in the dark should be going down because that means photosynthesis is happening. The one in the light should be going up. That means respiration is happening. Pretty simple. The next one's the most complicated one we have, I think, because it's essentially 2 experiments wrapped up into one. We're going to look at photosynthesis and see if things photosynthesize faster at warm temperatures or cold temperatures. So one of them is going to be in room temperature, just like this. One is in room temperature under a lamp. Another one's going to be in an ice bath. Make sense? So again, you have 2 of them that you're going to put in the light for photosynthesis, 2 of them that you're going to put in the dark for respiration. So right here, where are we at? Room temperature should increase, yeah. So these 2, when we're looking for an increase, that's photosynthesis, right? We're looking for an increase in photosynthesis. These 2 are for respiration. We're looking for a decrease. Again, for the photosynthesis, both of them are in the light. For the respiration, both of them are in the dark. For the photosynthesis, one of them is at room temp, and the other one's at ice. For respiration, one of them is at room temp, one of them is at ice. Everybody make sure you understand that, right? Is everybody clear? It's basically 2 different experiments wrapped up into one. How is photosynthesis affected by temperature? How is light affected by temperature? Also, when you write your hypotheses, this is a good time to remind you, it'll ask you why. So what's your logic behind? Like, do you think temperature has an effect, right? So you're going to answer why. So when you answer the why part, remember we have separate experiments. So for example, when you write your hypothesis to this, forget about the other experiment. So you don't want to say something like, I think the warmer the temperature, the more it'll photosynthesize and respire because there's more light intensity. And that's where you went wrong, because light intensity was the first experiment. Does that make sense? So your reasoning for any of your hypotheses should have nothing to do with the other experiments. We have specific experiments that deal with light intensity. We have specific experiments that deal with light versus dark. We have this specific experiment that deals with temperature. And then the last one, as we're about to talk about, deals with different light color. So don't mix your hypotheses. Does that make sense? Okay. Anyway, the last one. Again, we're looking at wavelength. Does different color of light affect the rate of photosynthesis? For all 4 of these, we're expecting it to increase. Let me talk about this really quickly. What color shirt is his? What would you say his shirt color is? Sorry, let me touch it. Mostly, what color is his shirt?
Red.
Red, right? And why does that look red to you? The reason it looks red to you is because white light is hitting it, and white light is all the colors, and it's reflecting red back to you. Does that make sense? So his shirt absorbing all the colors except red. So when you see his shirt, you're looking at the one color it can absorb. Mine is not absorbing blue. So what color, when you think of a plant, usually what color do you think of? Green. Meaning plants absorb all colors except what? Green, right? That's the one color that we're reflecting. So if you had to guess, based on this list right here, which one of those do you think is worst for plants? White, green, red, or blue? Based on that information. The worst, actually, let's start with the white one. Excuse me. Let's start with the best one. Which one of these do you think is the best? White, green, red, or blue? White, good. Any guesses why? Should be white, anyway. Like I just said, white color is all the colors. So it doesn't matter if blue's the best, or red's the best, or green's the best, whatever. They're all combined with white, so white is therefore the best. That being said, going back to what we were talking about earlier, plants are green, so what color do you think is the worst for them to grow in? Green, good, because they are green because that is the one color they're not absorbing. They're absorbing every single color other than green. So the fact that it's bouncing green back to your eyeballs means it does a really bad job of absorbing green. In theory, but we'll see. Any questions about that? All right, moving forward. Let me make this very clear. And if you've done the pre-lab, well, I haven't graded it yet, so that's the good news. If you've done the pre-lab and got a different answer for this, you can go change it now. The purpose of these experiments is not to see how pH changes. Like the goal isn't like, all right, let's see how light intensity changes pH. Let's see how temperature changes pH. No, we're just using pH as a measurement. Does that make sense? pH isn't the end goal here. We're just using pH to measure photosynthesis and respiration. And like we've already talked about earlier, there's other ways that you could set this up, right? We could do other ways of measuring photosynthesis and respiration, but our options are limited with a 2-hour lab. Also, keep that in mind too. How else could we measure? So at the end of your lab report, and I'm going to come back to this in a second, at the end of your lab report, you need at least a paragraph about that. So here's the experiment we did. You're going to write about whichever one of the 4 experiments you did. You're going to write about it. You talk about your outcomes, and then you're going to have a paragraph where you talk about what else could we have done. Hey, this is how we test our hypothesis, but how else could we do it? And I've already given one example. Like I said, we could grow a plant and measure its height, right? If we had more than 2 hours, we could do that. So keep that in mind as you're going through your experiments. These are just some random notes, some things to consider. No cuvette in the dark should increase the pH, right? It's in the dark. That means it shouldn't be getting any light. It shouldn't be doing photosynthesis. Therefore, the pH should not increase. If it does increase, that means there's something wrong, right? There must be something wrong with either the pH indicator or something you're doing, and that would invalidate your results. Does that make sense? Something clearly went wrong if your pH is going up. And we'll talk about that if it happens. There are some exceptions. So yes, that's also a question. I think after every single experiment, that's a question. Your results are invalid if that happens. Anyway, any questions about that? All right. While you're performing these experiments, think about how they can be performed in a better manner. Always, this is a common thread, right? Throughout the semester, I've been saying that. Like, how can we make this better? Like, for example, we're only doing the colors. We're doing red, green, blue, and white, right? So this is one example. What if we did more colors, right? Or what if we did it more than once, right? And we averaged our results. There's so many different ways. So think about that. And as we're doing the experiments, as usual, he and I will be talking about it and say, hey, this would be better if we could do this, or if we could do that. Anyway, any questions about that? All right. How to write a lab report, really quickly. Now, let me say this. Before I jump into this, I'm going to try to go through it quickly. First of all, do not look up how to write a lab report. Writing a lab report is a lot like submitting something to a scientific journal. Every journal has a different way they want it done. So if you look up how to write a lab report, you're going to find a million different ways. But you need to do it the way we want it done. Does that make sense? The way we're about to teach you, that's the way to do it. So do not Google how to write a lab report. Follow the directions we're about to give you. Another thing to mention too is this isn't a test. Like I'm not like saying, go write your lab report and report back to me. You can have us help you. We can meet you online and I can walk you through it, we can talk about what you're going to write before you write it. That way you don't waste your time writing something you don't need to or stuff like that. So let us know if you need help. But here we go. First thing you might want to know is why, right? I think there's only one biology major in here. I don't think that person's here today anyway. But anyway, so as a non-biology major, why do I have to write a lab report? It makes no sense. I'm a music major. I'm a business major. I'm an education major, whatever. So why? Well, first of all, it's part of the course. I don't make the rules. Someone else told me this has to be done, right? So for every other reason, that's the most important. I didn't come up with this. You have to write a lab report. So past that, why does it matter? Well, you have to truly understand the experiment to write a lab report. Anybody, well, not anybody, but almost anybody can go through the motions, write down the data, answer the questions about the data, right? But to truly understand the experiment, or you have to truly understand the experiment to write a lab report. In a perfect world, I would have you write a lab report for every lab instead of doing those worksheets. But that would take way too much time out of your day. Again, you're not biology majors, so I wouldn't expect you to do that. Even we didn't have to. Did you do that as an undergrad? Like a lab report for every lab in biology? We didn't either. Not in biology. We did in some chemistry classes. Anyway, Another thing about this is that help you understand research articles. Because once you understand how to write a lab report, which again is different depending on your source, same thing with research articles, right? So once you have this idea of how papers work, I'll do that. Whew. Okay, like I said, do not use the internet. And what I mean, I'll put a little star on that to remind you again, don't look up how to write a lab report. We're going to tell you how to write a lab report. Don't do anything that the internet says. Now, you can look it up for other reasons. Like you're going to do some research on the different colors of light or different temperatures, how it affects respiration, whatever. You can look that stuff up, but whatever. Only write about the experiment that you did. Good there, right? Oh, excuse me. And by the way, I'll make this available to you. So this whole PowerPoint, I'll give you access to it. So yeah, you're going to do one experiment. You're going to be responsible for all the data. You're going to answer questions for all the experiments. When you write your lab report, you're going to write about that one experiment that you did. It's always going to be past tense and first person. So say, I did this, I did that, then I did this. So be careful when, and we'll jump back to this, but there's a part called materials and methods. This is usually where people screw this part up because they'll copy it directly from the lab manual, but they shouldn't. So they'll say, first, take this cuvette and put it 10 centimeters from here. But you don't want to do that. Just past tense, first person. First, I did this. Then I did that. Double space it. What else? And all this, again, you're going to be given all this information. I'm just giving it to you now so we can talk about it, but this will also be given to you in a document. There is no minimum as far as words or pages. And as far as I'm concerned, the shorter the better. Your idea, this isn't independent work. The longer it is, the more points you get. So you don't need to be descriptive, right? This is a lab report. This isn't creative writing. You don't need to say something like, I gingerly grabbed the cuvette of beads, and placed it oh so gently under the thing. And I waited patiently for 5 minutes before I proceeded, right? You don't want to talk like that. Just, I did this, I did that, to the point. That make sense? Just get to the point. You're not trying to make it particularly long. You're going to turn it in the same way you do labs and independent work, in that there's going to be a document. I'm going to provide you with a document, and you're going to share it. That makes sense. The same way you go to a lab, you make yourself a copy of it, and then you share that document with me. That's what's going to happen here. I'm going to share a document with you. You make your own, you share it with us. That's how you're going to write. Good? All right. You have 2 weeks from the day you get your lab back. That's your due date. So let's say, for example, you get your lab done next Monday, and then you write on it, and by Tuesday you say, hey, I'm done. Can you grade it? Wednesday, I grade it. You get your grade back on Wednesday. Then you have 2 weeks as of that day to finish your lab report. Make sense? 2 weeks from the day that I give you your grade back. And that'll be part of the grade. So I should let you know that too, that you're also grading. When you grade this lab and you give them the grade, then also mention, give them the date. So 2 weeks from today, whatever date your lab report's due. All right. Anyway, that being said, you don't have to wait that long to start. You don't have to wait till you get your grade back to start your lab report. You'll see why as we go through this. Only some of the lab report is actually required. You only have to do, how do I put this? You can write a lot of your lab report without even doing the lab. There's only some parts of the lab report that you need the lab for. We'll talk about that when we get there. All right. As always, this should be free of grammatical, spelling, and typing errors. I'm not really picky with that. Look, here's the thing. You're writing it on Google Docs. So if you screw up, there'll be a big red line. Just fix it. Does that make sense? Like usually that's how I grade. I don't usually, I'm not nitpicking. But if Google highlights and says to me that's incorrect, I'm like, yeah, that is correct. Incorrect. So mostly if Google didn't notice it, I probably won't notice it either. It should make sense. So if you want to, if you have anybody, you should be able to explain your experiment in enough detail where somebody could understand what you're saying, right? You should give them enough information, and we'll come back to that, in your lab report, where someone who's never had this class can read it and understand what you did and what your results are. And this kind of goes in there too, accurate and correct. People do mess this up. You will lose points for this. This is one of the bigger mistakes. It's like when someone's describing their data, they're saying, my hypothesis was incorrect, but then the hypothesis was supported by the data. But, well, that's not accurate, so you lose points for that. So just make sure you're very careful with the things you write. Make sure you're saying the correct things. It should be precise. We'll cross that bridge when we get there. Succinct, I already said that, right? So shorter is better. You're not trying to be entertaining. You need any kind of like descriptive language. Oh, and the big thing, it should be clear that you understood the experiment. I guess point-wise, that's not too big, but this is really what I'm looking for. After reading your lab report, I'd be like, they had definitely not understood what they did, but they understood what their results mean. All right, so I'm going to give you a rubric, a grading rubric. Like I already said, you're going to have a document, you're going to make yourself a copy of that document, then you're going to share it with us. And this is what's going to be on that document. So it's really hard to screw this up. If you just follow exactly what it says, you should be okay. You're going to need a title. You get a point for that. Make sure your title is accurate. So for example, your title should not say photosynthesis respiration lab report. Be specific and say, what experiment you did. How temperature affects photosynthesis and respiration. How wavelength affects photosynthesis. Things like that. So that's the first section. Next, you're going to have an abstract. An abstract is basically like a brief description of what the whole lab report's about, right? So you're going to have to title it. In your paper, it'll say abstract. You get a point just for giving it a title. Again, like I said, it's going to be a brief write-up about what this whole lab report's about. It's going to include a basic summary. So you say, in this experiment, I wanted to see how light intensity affected respiration, and I found out that, being 15 centimeters from the light was the best, or whatever it may be, right? Just give the basics. You don't explain how you got your information. You just say, this is the basic results. Later, you would get into more detail, but in the abstract, this is what we found out. Here's what we're trying to figure out. Here's what we found out. And then finally, you'll see this on every section. Does it include anything that should not be in this section, right? So again, only the stuff in the abstract should be in the abstract. You don't want to give details, for example, of how you did the experiment. First thing we did was put 1 cuvette 10 centimeters from the light, or first thing I did was put 1 cuvette here, 1 cuvette here. You don't want to talk about that in the abstract. The abstract is just the very basics. Any questions so far? Oh yeah, I'll come back to that, sorry. So here's the sections you're going to need. These are the sections of your lab report. Each one's going to have a title. Each one's going to be labeled. So you have a title that we just talked about. You have an abstract that we just talked about, followed by an introduction, followed by materials and methods, followed by results, followed by discussion, followed by figures and tables, and then finally literature side. We're going to talk about all these very quickly because, again, you're going to have access to this, so there's no need to beat a dead horse here. Already talked about that, how to title it, right? Abstract, already talked about that. Introduction, here we go. This is important. And again, I'm just telling you this. You're going to have access to it. The only reason I'm even telling you is that way if something doesn't make sense to me, you can ask now as opposed to doing it later. Introduction. This section gives enough information that the reader can understand the rest of the lab report. For example, at the end of somewhere in your lab report, you're going to say, the pH of this thing was 6. pH of this thing was 7. The pH of this thing was 8, whatever, right? You're going to say that. And that makes no sense. Here we are talking about photosynthesis and respiration, and you're talking about pH. This is what the introduction is for. You should give enough information to where if somebody reads something about increasing pH, they'll know what you mean. First of all, it should be labeled just like everything else. It'll literally say on your paper, introduction, and then you'll write your introduction. Oops. It should give enough information about photosynthesis and respiration that the reader will understand the rest of the lab. Right? So again, going back to this whole idea of pH changing, at the very least, you should say that photosynthesis uses carbon dioxide and carbon dioxide and water to produce glucose. And then you should, at the very least, mention respiration produces carbon dioxide, right? That's very important information because without that information, you can't explain the pH thing. If you can't explain the pH thing, then your results don't make any sense to anybody. It should include the question you were answering in the experiment. Yes, I already know you put that in the abstract. This is a good time for me to tell you some of this is redundant. That's on purpose. That's normal. So yes, once again, you'll say that, I wanted to see if light intensity affected the rate of photosynthesis, right? You will repeat the question. Then again, you'll give your hypothesis. Whoops. Include your hypothesis and why I chose it. So again, I wanted to see if light intensity affected respiration, photosynthesis. I hypothesized that the The more light there was, the greater the intensity. I chose this because I know that photosynthetic organisms need light for photosynthesis. Whatever, right? Include your hypothesis. Include a sentence of what made you think of that hypothesis. This is really messed up. There we go. And then finally, the last part of your introduction should explain how photosynthesis and respiration affects the pH, right? So that whole thing I told you about earlier, you're going to want to explain it too. Photosynthesis uses carbon dioxide, therefore there's less carbon dioxide in the water. Therefore there's less acid in the water, therefore pH increases. Respiration produces carbon dioxide, therefore there's more carbon dioxide in the water. Therefore there's more acid. Therefore, when pH goes down, photosynthesis is happening. Any questions about all this? All right. It should not include a lot of things, right? As usual, you want to make sure that you don't have anything that shouldn't belong there. One of the biggest mistakes I see here are people going in materials and methods. They'll start talking about how they did the experiment, but that's not what the introduction is about, right? That's later. Or the results. You don't talk about the results in the introduction. As a matter of fact, you know what might help, and I mentioned this earlier. Like I said, you can start writing your lab report before you do your lab. If you have the ability and the time, I recommend doing your introduction now, right? Your introduction is not what you did, it's just what you're going to do, right? So this is all the information, just the background information. So you really can't do the introduction before you do the rest of the lab. And it might help you with the rest of the lab. Any questions about the introduction? Materials and methods is very straightforward. Basically, you're just going to write enough to where somebody could read your lab report and reproduce your experiment. Does that make sense? You should have enough information about what you did where someone can read it and redo what you did. It really is that simple. But really quickly, we'll go through this. It should include everything used, right? All the material used. For example, a lot of people forget to mention the fact that you're using algae beads. They'll just say we had a cuvette. And to remind you, these things are called cuvettes. But yeah, these aren't just cuvettes. There's water in there. There's pH indicator in there. There's algae beads in there, right? So make sure you mention everything. And just as usual, it's a good time to mention this too. If you screw any of this up, as usual, one of us will leave a comment and say, hey, don't forget this. And you can fix it. If it's before the due date, you can fix it for full credit. If it's after, you just get some of the points back. Point being, there's no pressure, right? Even if you screw things up, you can still make up for it. It should include not only everything used, but everything you've done, right? So make sure you mention everything. Like I said, someone should be able to read it and reproduce your experiment. Do not copy it word for word from the lab manual, right? For example, this is first person past tense, but your lab manual, it's not. It says first do this, first do that. So don't copy it from your lab manual. Remember, it's first person past tense. Here's another big one. If you change anything in your experiment, that's completely fine. For example, this first experiment, you're supposed to be 10 centimeters, 15 centimeters, and then 25 centimeters from the light, like I showed you over there. You can change it out. I don't care. This is your experiment. You can change these if you want. Make them closer, make them farther, whatever you want. But whatever it is that you actually do, make sure that's what you write. Does that make sense? So if the lab manual says, put them at 10, 15, and 25 centimeters, and you say, nah, let's not do that. Let's do 5, 20, and 30 for some reason. That's okay. You can do that. Just make sure you write down what you actually did, not what the lab manual tells you to do. Does that make sense? All right. Because you do have a little bit of freedom. Now, granted, I would recommend talking to one of us before you go making a big change, but it's your experiments. It should not explain why are you recording the pH, right? So yes, again, materials and methods, you're going to say every 5 minutes I recorded the pH. You don't have to say why you're doing that. You've already explained that in the introduction. So again, I know some of these things seem redundant, but some of them aren't, right? You have that information in the in the introduction, so you don't have to explain it here. You can say, here's what I did. You don't have to talk about why you did it. Just here's why I did. Here's what I did. So any questions about materials and methods? All right, the next two sections you have been trained to do, and you might not know. I've been doing this on purpose with your lab worksheets. If you'll notice, after every table, for the most part, you have a part where you just have to restate your results, right? There's the results in the table. And then the next question seems redundant, like you just have to write the whole thing again, right? So you just say what's on the table. That's what your results section is. And then later I ask you to compare your results to your hypothesis, right? So then there's a question after that where I'm like, hey, what was your hypothesis? Was it correct or not? This was all on purpose. You are now ready to do this. You've already been trained to do this. So let's talk about it really quickly. The results section. It should simply put the data in words. So yes, everything that's going to be here on the table, you're just going to put it in paragraph form. The cuvette and closest to the light started at a pH of blank and ended at blank. The cuvette here started at this pH and you're just going to put it in words. Does that make sense? Just put it into words. That's all. It should include the starting and ending pH of all the cuvettes. It should include which cuvettes increase the quickest. I wish I wouldn't have erased that earlier. Because remember, I had that one example where they both started at 4 and ended at 7. So then we had to look at the data in between. So you're going to mention that too, right? If one of them happened to happen quicker or slower, then you mention that. There we go. Oh yeah, if something didn't change, you mention that too, right? This cuvette started here, this one started here. This one didn't change, right? You'll just mention that. You don't talk about why it didn't change, you just say this one didn't change, right? Again, with all of this, you don't explain what it means. You don't say this one went at this pH to this pH, meaning it photosynthesized. You don't need to do that. You just say it went from this to this. Finally, this. Identify the cuvette by their condition. So yeah, you might write numbers on here, like this one might be number 1, number 2, number 3. But when you're writing about it, you don't say cuvette number 1 started here, ended here, cuvette number 2 started here, ended here. You describe what its condition was. So again, the cuvette closest to the light started here, ended here. The cuvette in the dark started here, ended here. That makes sense? Because again, it needs to make sense to somebody who's reading this who didn't participate in the lab. So they won't know what it means by cuvette number 1 or cuvette number 2. We're getting there, guys. Again, I know this sounds weird, but in the results section, the word photosynthesis and respiration never come up once. This is just straight up data. You're just telling us what happened to the pH. The next section is when you decipher this. You explain what it means that the pH went from this to this. But in the results section, you don't mention photosynthesis, you don't mention respiration, just straight up talking about the results. Any questions? All right. Like I said, the next section is where you give some context. That's the discussion. First thing you're going to need to do, and I know this sounds redundant, but again, you've been trained to do this with the questions you've had on your lab worksheets. You're going to need to restate the purpose of the experiment. So you'll say again, in this experiment, I wanted to see, in this case, how light intensity affected respiration, or photosynthesis, right? You'll readdress it. But this time, this time you also readdress the results. So again, I know it seems redundant. In the section before that, you just said this pH started here, ended here. This pH started here, ended here. You're going to do that again. Except this time, when you do it, you're going to explain what it means. This pH went from here to here, meaning it photosynthesized. This pH went from here to here, meaning it photosynthesized even faster. This one went from here to here, right? So you explain what the results mean. You repeat it, but this time you explain it. Yeah. So any questions so far? I know it seems redundant, but again, you're going to write what you wrote in the results section, but this time telling what it means. And not only are you explaining what it means, but while you're at it, you are then addressing your hypothesis. And then once again, I know this seems redundant, but you've been trained to do it. You're going to restate your hypothesis. So I wanted to see how light intensity affected the rate of respiration. My hypothesis was this, and my results support it, as you can see, and then repeat the stuff that supports it. Or you can say my hypothesis does not support it, as you can see by, and then use the results to explain it. Yeah. Now, if the data does not support your hypothesis, then you'll need to write about that. Like, what do you think went wrong? Was there something wrong with your hypothesis? Was there something wrong with the experiment? Right, you'll talk about that there. And also, just so you know, I have to point this out, and I've done it all semester. I'll do it all semester. This experiment that you're doing does not prove anything, right? So don't use those words in your lab report. According to my result, or my results proved that my hypothesis was correct. No, it did not. It may have supported your hypothesis, but this is not proof. Any questions so far? All right, after that, then you need to address the limitations of the experiment. Again, you've been trained to do this. Every lab we do, we talk about that. What could we do different? What's wrong with this experiment setup? How can we do things better? For example, if the results suggest that algae photosynthesize better at a more intense light, right? Let's say you do this experiment and you find out like this one closest to the light photosynthesize the best. Does that mean that that's always going to be the case, right? If we were to do one at 5 centimeters, does that mean it would be even better? So you just write about like the limitations of your, not just your experiments, but also the limitations of your results. Any questions about that? All right, and then finally, like we've already said, at the end of your discussion, you should write about other experiments that you can do. Like, all right, here's how I tested my hypothesis this time. And then you could write, but here's other ways I could write test my hypothesis. Because in real science, that's what you do. You don't just do an experiment once, like that supported my hypothesis. I told you so. No, you go back and you think about other ways to test your hypothesis. Except in this case, you guys are lucky. As much as you might hate writing a lab report, here you only have to write about what you have to do next. In real life, you would actually have to do those experiments. Here, you just write about it. Any questions about that? All right, figures and tables. This one's so simple. So simple, especially now that we're doing everything digitally. Figures and tables, you're basically going to take what's on your lab worksheet and copy and paste it here. That's it. You're going to need a table, like you're going to fill out already on the worksheet. You're going to make a graph, like you've been doing all semester. The table is the easiest part, because again, it's what's in your lab worksheet, right? So you can literally copy it and paste it, and you're done. You do want to label it. You'll use the words table 1 and then a brief description. So table 1, change in pH of cuvettes in different lighting intensities, right? You just describe the information in the table in the basic sentence. Any questions about that? And again, don't forget your units. We're using pH, so you're good there. Graph, again, you already know how to use a graph. You could use Excel or Google Docs. This is the one time that you can't do it by hand. So you're going to need to make an actual graph. You're using a computer, make a graph. I can help you. He can help you. Tutors can help you. If you need help doing it, let somebody know. It can be done. As always, you're going to label it. So figure 1, and then describe, use a title that will describe the graph. Make sure that the title describes the data. So again, a horrible graph title would be photosynthesis experiment. What does that mean? Be more specific. As always, it should include enough information that the reader knows what's on the Y and X axes. Again, as always, your graph should make sense to somebody who's never read the lab and was never in lab. There should be enough information where someone can read it like, oh, I see what happens. I see what this is saying. Yeah, that's it. Any questions about the graph or the table for that matter? All right. The really, really easy section, and I'm only including it because it's just kind of required. Almost every type of lab report requires it to sort of line. Literature cited, just to list your sources. In your case, and this sounds really contradictory because you know how big I am on sources. In this case, it doesn't matter. Your sources can simply be the lab worksheet, and your textbook. It can be that easy. If that's all you use, then that's all you have to list. And also, I'm not picky, just like with independent work. I don't care if you use APA or MLA. I don't care what it is. As long as you have a literature-cited source and you literally list your sources, that's it. Now, if you do use a source outside of your lab manual or your textbook, then you do need to list it. But just like independent work, you can just give me a link and that'll be fine. And that'll be very important because I have had some people write something incorrect in the lab report, and then I check their sources, and their sources backed them up, so they didn't lose points. Now I had to tell them that their source was incorrect, and that was wrong information, but they didn't lose the points because they backed it up with the source. So any questions about literature cited? Okay, that's it. We are done for the day. Make sure you guys get your pre-labs done by Sunday. There's an extra credit lab where you can do this virtually. Oh, wait, is that correct? Yes, I think there is. If there's not, I'm going to fix it up and make sure there is one. If you do the extra credit lab first, you get more points.